Hi everyone, Rose Webster here. I'm going to update both petitions with this one video. 6 days ago, Massachusetts Environmental Trust responded to my post and asked me if I had sent my information to Woods Hole Oceanographic Institute. And I have sent my petition to every known Facebook group or whatever out there. But I did respond. I said, well, no, not him specifically, but I've let our Canadian politicians know, Dominic LeBlanc, Jane Philpott, the NOAA, National Oceanographic and Atmospheric Administration, and others know. Lots of whale groups are aware of my efforts, but have not been receptive at all. I guess I could try this gent too. And, you know, it's funny, I'm shot down in a lot of these groups. So I don't know if that's going to go anywhere. In fact, I'm just going to be blunt. If there are no calves next year, I'm pretty sure Wolbachia is the root cause of their infertility. So I'm going to have to move on to humans, which are becoming infected. However, physicians can't test for Wolbachia infections in a timely manner, at least according to Medscape, and that's what I'm trying to change here. So this was just mind-boggling. I read something about, Auburn, Alabama, it might be too early for it to be deemed a cluster. Well, guess what? The national incidence rate of uveal melanoma in the U.S. is 6 people per 1 million. In Auburn, Alabama, what we're up to now, with a population of 63,118, roughly around there, 31.6 people per year on average have been diagnosed, which is actually 83.5 times the national average. So if this isn't a cluster, can someone tell me what is? This is getting ridiculous, absolutely ridiculous. Now, I did have a lawyer reach out to me and kind of wondering if anyone could be at fault. Now, here's the key thing. Yes, Wolbachia is in the environment and it's in the insects. You cannot be testing waterways and plants and environments. It has to be on excised tissues within a timely manner, ideally within the first week, because bacterial counts and even intact Wolbachia can be found in the blood. And I suspect it'll be in lymphatic tissues like lymph nodes that get excised, etc., for a longer period of time. But the bacterial counts are actually 600 times that of the arthropod host, or the insect. Now keep in mind, there's about a dozen types of blood-sucking insects out there that transmit diseases. So we're not just talking ticks, which are associated with a rash and that eschar. We could be looking at other things, perhaps even airborne. Aedes aegypti don't live forever. These mosquitoes don't live forever. They decay. They live a few days to a few weeks. So this was my response to one law firm, is Mosquito-Mate is claiming in terms of the FAQ was, can it transmit disease? No, they say. Our ZAP mosquitoes are male mosquitoes, and male mosquitoes do not bite. Because they do not bite or blood feed, male mosquitoes do not transmit pathogens to humans, pets, or other animals. Well, here's what you're not being told, is Wolbachia-infected males mate with females. Some of these females will have viruses, and the presence of a virus facilitates the invasion of Wolbachia. And some of these females won't have viruses. Okay? And this is how Wolbachia gets into them. It's maternally inherited. So the females that do blood feed, they can transmit the Wolbachia along with any other viruses they might have or not have, along to other mammals, including humans. So it amazes me that this point is like lost. There's some kind of clever wording, or it's like people have put up blinders. They don't want to know any more than that. Now here's the thing. Wolbachia can survive at least a week in a dead host with no decrease in viability. And it's ample time for other organisms to acquire and spread it. And remember, when birds and bats die and fish die, these things are floating around. People aren't cleaning them up right away. So here we have other like areas of where this can keep on spreading, and it's maternally inherited. It's in the larvae, pupae, the flying mosquitoes, etc. And the thing is, this was known in 2006. But what made me almost fall off my chair is when I discovered that there was a 2003 study, it seems to be ignored, that bacterial growth of Wolbachia pipientis could be obtained in human embryonic lung fibroblast monolayer at 28 and 37 degrees Celsius. Now, they didn't convert Celsius to Fahrenheit. So if you're not paying too much attention, you might think, oh, that sounds cold. No, 37 degrees Celsius is 98.6 degrees Fahrenheit. That is normal human body temperature. And to continue the quote, confirming that its host cell range is broader than initially thought. So, what I said, it's going to take some guts and integrity on the part of law firms to pursue this. Gag clauses are only going to ensure many U.S. citizens will bury a child someday, and probably Canadians too, because for some reason, Dr. Theresa Tam, who studied some of this, but it is associated with the World Health Organization, it doesn't dawn on her that 4 million Canadians go to Florida per year, where these releases are ongoing right now until the end of July. It doesn't maybe dawn on her that these things are going to cross the border. So you're going to see them in Ontario and Quebec because it's just south of our border. These things blow over. 80s are very light. Wind carries them, waterways, etc. They don't live very long. They decay. The Wolbachia can live for at least a week inside them. So what we really have are time-release capsules with a reproductive parasite that is potentially cancer-causing as well, or what appears to be the clinical appearance of a cancer. But when I'm viewing some of these videos of people talking about their symptoms, I'm thinking, this sounds like an infection. They're describing lymph nodes swollen. They're describing a fever, which is, I think the cardinal symptom is a fever. Liver tests are coming back with elevated transaminase, I can't say the word, but elevated levels of ALT and AST in the liver. You don't have a lot to go on. You may not see that rash. That rash may not happen because if you have a mosquito or inhalation causing this infection, you're not dealing with the saliva of the tick that's associated with Wolbachia or rickettsial infections. And I'm sorry I'm not saying these terms correctly. Now, moving on. I could not believe this. I sent the National Federation of the Blind of Wisconsin my petition. And on Facebook, the person was not really receptive, wanted to know why I was getting a hold of them and so on. And they left me with this final 2 sentences. Heavy-duty antibiotics often can cause blindness also. Again, this petition seems to be from 2015. So obviously this person's not scrolling past my intro image where I put published 2015 Chendong et al. And those study points. And I couldn't believe it. I had to put it out there. I did tag ophthalmology groups that I know that would see it, and even the magazine Retinal Physician, where I looked into this. I thought this doesn't sound right. So what happened was in October 2013, two doctors, Peter A. Karth, that's with a K, and Theodore Lung, L-E-N-G, they're both MDs, and they have other degrees. What they did is they did a case control study of 4,384 patients with retinal detachments and 43,840 age-matched controls without retinal detachments. And what they found is that 3.3% of the retinal detachment group were taking antibiotics, and only 0.6% of the controls were taking medications. So the strange conclusion being drawn from this is that somehow the antibiotics are causing the retinal detachment. My question is, well, why were these people put on antibiotics? Because the primary infection, such as rickets, we'll say Wolbachia, could be the underlying undiagnosed reason for the retinal detachments. And yes, I know there are other reasons. There's other viruses and types of infections. I realize that. Even fungus can infect the eye. Now, physicians are currently unable to test for Wolbachia infections in a timely manner. I've said this before. That's what the petition's about. Another quote from this study, those subgroups that have recently taken but had discontinued the antibiotics as little as 1 day before being diagnosed were not statistically different from the controls. Animal studies have shown that antibiotics are cleared from the vitreous within 12 hours after intravitreal injection. Sorry, I have a problem with my jaw pronouncing things. And here's the key point that is lost, at least on this person and other people who chime in and agree with them. Although a mechanism by which antibiotics can cause retinal detachment may exist, a case control study cannot prove true causation or association, merely correlation. So my point is, citing this 2013 study as proof that antibiotics cause retinal detachment is highly misleading and dangerous, because for the past 30 plus years, the US has experimented with Wolbachia-infected Aedes releases. Wolbachia targets, depending on the strain, the optic lobe and retina. It competes with our mitochondria and targets, depending again on the strain and temperature even. So there's a lot involved to this. The brain, spinal cord, nerves, including nerves to the stomach and intestines, muscles, gonads, lungs, kidneys, heart, liver, eyes, ears, and or pancreas. There you go. There's a lot to this. Doctors aren't allowed to test for it. So I wanted to let you know that I think we're making an impact, because I also received an invitation. So I'm going to show you that now.
Okay, so yesterday I received this invitation from Alani Mitunho, and I don't know why they thought I was a doctor, because I don't have that anywhere written down, but anyways, they said, we invite you to participate in a research entitled Dengue Prevention Using Genetically Modified Mosquitoes, Current Facts, Literacy, and Public Opinions, coordinated by Professor Carla Sousa at the Institute of Hygiene and Tropical Medicine of the Nova University of Lisbon. And I think that's in Portugal. Therefore, we solicited your participation in a semi-structured interview that could be done by Skype. The topic in debate is about the use of a modified insects for vector-borne disease prevention. Or if you agree to participate, we would like to schedule in May. As your preference, as soon as possible, let us know. Okay, so I'm responding very quickly to them then, and I'll show you that. I'm not going to read it all out, but I'll give you some snippets here. So I thank them. First of all, I'm not a doctor. My background, nursing, orthotics, prosthetics, ophthalmology, technical imaging, and assisting. Those who've employed me know I always ask questions. I go to the continuing education. So, I'm not somebody that gets a title and then just sits back, because I don't know, I'm insanely curious. My father was an environmentalist engineer, so maybe the engineer part explains why I'm obsessed with solving things. But anyways, so I said what my work includes, and I'm working on a documentary. So read the use of modified insects for vector-borne diseases prevention. Well, you have not clarified the method you're hoping to discuss with me. And I believe there are two main methods. There might be another one. I heard about sterilizing mosquitoes, but I don't know enough about it. The 2-gene self-limiting modification of Aedes mosquitoes by Ozotec, and the much more worrisome Wolbachia-infected Aedes releases carried out by the World Mosquito Program, which used to be known as Eliminate Dengue, and Mosquito Mate, which is based out of Kentucky. And those two groups have been working on it about 30 or more years. I said here, I'm not a fan of either technology, but I'm far less concerned about Ozotec's method than I am about Wolbachia-infected Aedes. And I go on about how safety tests were never carried out on vertebrate species. It's akin to putting genetic dynamite in a species. It can survive at least a week in a dead host, and lateral transfers to other species have happened. In fact, they are common. I agree with Dr. James, who, quote, suggested that these genetic tools might not be the best strategies for Zika, and I'll add dengue or other viruses, given that at this point there seems to be multiple vectors, not only at the species, but also at the population level. The current genetic technologies would not be appropriately applied to such complex systems. And I completely agree with that, but there's a huge effort to downplay what's going on with the Kulik species. And Kuliks are almost opposite to Aedes. I'm just going to say this. They breed in filthy water. They're highly competent vectors, some of them, of encephalitis viruses, which Zika is, along with Japanese encephalitis virus and other ones. So they're overnight active. And you're always told daytime active. Every time I hear daytime active are all you got to worry about, I cringe. And here's the thing, Ozatec even made crucial points on a docket. The potential genetic modification that could result from the use of Wolbachia pipientis is wholly undefined and could introduce over 1,000 new genes into the target mosquito with unknown consequences. So you've got to weigh a 2-gene modification that's self-limiting, 2 well-studied genes against 1,000 new genes that are put into a target mosquito, again, with unknown consequences. And they've used several strains. And again, I highlight something that should be making the rounds in every oncology department in North America and around the world, the 2015 paper by Chen Dong et al. Wolbachia can infect mammalian cells, even human cells in vitro. Horizontal transmission in insects and among helminths occur via cell-to-cell invasion, predation, and cannibalism, among other possibilities, establishing the potential for horizontal transfer to animals and humans as well. Hence, Wolbachia should be further evaluated as causes of human infection. Now, I'll let you read through this because it'll be quicker for you. Of course, you can always pause and read at your own pace. So here's the thing. According to Professor Carla Sousa's biography, she acted as a consultant for Madeira health authorities during dengue outbreak and as a WHO consultant during the Mozambique's dengue outbreak in 2014. Now, this is why I want to be blunt with them about my position, because I know the gag clauses with the WHO are tight, and anything the WHO endorses or invests in, you really cannot say anything negative about it. That's how tight those gag clauses are and the funding clauses. So the bottom line is I believe Wolbachia-infected Aedes are causing the Kulik species of mosquitoes to become better vectors of viruses such as Zika, dengue, and others. And there has been a concerted effort by public health authorities, 4 governments, possibly more, in my opinion, our Canadian government as well, and extremely wealthy investors to hide this. The evidence by Graham and more recently Dr. Constancia Ayers should never have been ignored and or downplayed. Quote, Graham, who first demonstrated the transmission of dengue by mosquitoes in 1902 to 1903, thought that Culex quinquefasciatus, formerly Culex fatigans, was the vector. The current problem in the scientific community is groupthink, funding clauses, and greed. The studies that should be funded are not being funded. If you still wish to proceed with a Skype interview with me, 1, I will also be recording the session and possibly publishing it. Why? Because the media has edited other researchers like our Canadian Dr. Fiona Hunter, and I included that screenshot. 2, my position is that Wolbachia-infected Aedes are causing grave illnesses in vertebrate species, including humans, and that includes North Atlantic right whales, roseate terns. There's lots of shorebirds that are dying. Arctic terns, which would carry this worldwide when they die, of course. They've had breeding failures, and Adelie penguins have had a couple of mass breeding failures in the last 7 or 8 years. So it should never have been allowed to proceed. 3, the continuation of Wolbachia-infected Aedes around the globe is in fact a crime against humanity. I look forward to hearing from you. Truthfully, Rose Webster, Humanitarian Activist. So there you go. I'll let you know.